Hello, everybody, and welcome to today's Revive webinar from the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I am hosting this webinar on probability of target attainment analyses for dose selection in antimicrobial drug development. For those of you who are joining our webinars for the first time, Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All webinars are recorded in full and can be viewed after the live broadcast on our website, revive.gardp.org/webinars. I encourage you all to visit the Revive website to stay up to date about future webinars and watch recordings of previous ones. Right now, you can already register for our 4 next webinars. As always, today's presentation will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window in your webinar control panel, as shown on this slide. We will address the questions after the presentation, and we'll do our best to respond to as many as possible. Today's speaker is Shampa from the Center of Excellence in Infectious Disease Research at the University of Liverpool. The webinar will be moderated by Vincent from the University of Houston. Welcome, Vincent. I now hand over to you for a brief introduction.
Thank you, Victor. Good afternoon, everybody, or good morning, or good evening, wherever you are. I understand this is being broadcast to many countries. We are so glad that you can join us this afternoon, and it's my pleasure for me to introduce the speakers today. Dr. Shampa is a senior lecturer at the University of Liverpool and a PTPD consultant. She has 17 years of experience within the pharmaceutical industry. She has extensive expertise in using PKPD for developing clinical pharmacology and dosing strategies to support the clinical development and regulatory approval of new anti-infective drugs. Shampa is a senior leader within the antimicrobial pharmacodynamics and therapeutics group, which supports biotechnology and pharmaceutical companies to develop new antimicrobial agents by developing the critical PKPD package required for dose selection. As a consultant, the DAS also uses PKPD to inform critical decisions on adult and pediatric dosing to companies and funders, including CAPEX and Wellcome Trust. She has recently joined the UCAS steering committee as a PKPD expert. We are very glad that Dr. Jas can share with us her experience this morning. Shampa, now you can start your presentation.
Thank you, Vincent. Sorry, I'm just taking the controls. So as introduced, I'll be talking about the probability of target attainment analyses for dose selection in antimicrobial drug development. So I want to start off by focusing on what we mean by PTA, and I'll break this down for a minute and first start off talking about the target in probability of target attainment. So in antimicrobial drug development especially, we use preclinical animal and in vitro models to understand an exposure target. So we believe that by achieving this exposure in humans, this should be associated with an increased probability of clinical success. Now, in this field particularly, we believe there is very good and strong translation of preclinical efficacy data to humans. You know, the drugs that we're developing are directly acting on the bacteria. What we're measuring in the preclinical models is impact on bacterial growth or bacterial kill. And if we can get the right exposure to the right site of action, we believe that will translate very nicely. So to move on to the other part of probability of target attainment, focusing on probability, one thing we know is that there will be variability in drug exposure in humans. So it's not sufficient to just achieve an average exposure in humans that matches that exposure target that we're trying to achieve. What we want to do is ensure that the majority of population will have sufficient exposure. So that means what we want to have is a dose that has a high probability of target attainment. So what do we need to calculate PTA? We need to understand the relationship between drug exposure. So drug exposure is the pharmacokinetics, what the body does to the drug, and relate that to response, so the pharmacodynamics, what the drug is doing to the body. And here, when we're talking about pharmacodynamics, we're talking about impact on bacterial growth or bacterial killing. So what we need is a well-characterized PK/PD relationship, and that's going to derive a PK/PD target. So that target exposure that's going to be associated with that impact on bacterial growth. We also need to understand the MIC distribution of the drug, and we need to understand human pharmacokinetics. So we need a human population pharmacokinetic model. However, even if we have all of that, our PTA analysis is only meaningful if we have confidence in the data from which it is calculated. Now, some of these concepts, such as the PK/PD target, have been covered in previous webinars, but because it's so important to the analysis of PTA, I want to just briefly go over some of these again. So let's focus firstly on the PK/PD target. So what we're doing here is studying that relationship between exposure of drug and bacterial burden. So when we first start in preclinical experiments, we tend to look at impact of increases doses. So we quite often use gold standard models such as the neutropenic mouse thigh infection model or mouse lung infection model, and try and build a relationship between dose and effect on bacterial burden. And then we develop a relationship that's going to look something like this. And we want to select a dose that's going to maximize efficacy. However, we can't translate this dose directly from a mouse, which is dosed in mg/kg, to a human, because we know that the way the drug is handled in the mouse is going to be very different from human. So what we want to do is understand this dose in terms of drug exposure. So for each dose, we can generate a concentration time profile. So we can describe the pharmacokinetics of the dose. However, what we want to do is translate all of this concentration time profile into a parameter that we can easily use to set a target. So we can express various exposure parameters, and the ones we tend to use in this field to set an exposure target can be Cmax, the maximum concentration, the whole of that area under the concentration time curve, or time above a threshold concentration. And what we do is we integrate these PK parameters with our potency measure, which is MIC. So we look at Cmax over MIC, AUC over MIC, or time above MIC. It's essential for us to know which one of these parameters would best describe the data. So we're trying to find out how exposure drives the response. So the way that we do this is we conduct a dose fractionation study. So here we give the same total daily dose, but we give it in different ways. So we give that same total daily dose either as a single dose or the same dose fractionated, given half doses twice, or maybe a third of the dose 3 times. Then we take the data emerging from that and plot out exactly the same data, but express the dose in terms of our PK parameters. So AUC over MIC, Cmax over MIC, or time above MIC. Now, in this example here, you can see that there's no relationship. There's a complete scatter of data here, where the same data is plotted against AUC over MIC, and same with peak over MIC. But here with time above MIC, you can see a nice correlation with increasing time above MIC and a decrease in bacterial burden. So what this experiment has done is helped to define a PK/PD index. It tells us which of these parameters best describes the data. But this PK/PD index is generally derived from studying only a small number of strains, maybe 1 or 2. To really understand the magnitude of that index, so the percentage of time above MIC you want to maintain, or the number of AUC over MIC that you want to achieve, you need to understand this from an expanded number of strains. So in this example here, all of the different colors and shapes represent a different strain. And we're building a relationship between understanding what magnitude of time above in this example is associated with parameters such as stasis, so prevention of bacterial growth, or bacterial killing. And we tend to use the parameters of 1 log kill and 2 log kill. So there's lots of things we need to take into account when considering that PK/PD target. We need to consider the number of strains that we're using, the strains being relevant, the variability that we're going to get from these models, which models we use, where that exposure is being measured, and the emergence of resistance. And I'll go through these in a bit more detail in the next few slides. So when we think about the number of strains, we've got to think not only about the number of strains that we use, but then being relevant strains, so ideally clinical strains. We want to choose strains that should encompass the range of MICs that we're developing this drug to target. And ideally, you want to include particularly several MICs towards the upper range of that target MIC. So what I'm saying here is that there's no golden rule in how many strains you should study, but certainly it should be more than 2 or 3. Another thing to think about here is not only number of strains in totality, but consider the number of strains for each pathogen of interest. So you may be developing a drug that's quite broad spectrum and it's going to target both Enterobacteriaceae, Pseudomonas, Acinetobacter. You want to ensure that you select a sufficient number of strains that's going to describe each of these pathogens, because certainly that PK/PD target might differ across the different pathogens. Another thing to keep in mind is studying both wild type and resistant strains so that you're really understanding the PK/PD of what you're trying to target and ensuring this doesn't differ between the two. What this graph is showing is a combination of data from E. coli wild type, E. coli non-wild type, so expressing ESBLs, and in the blue Klebsiella wild type, and then the filled-in squares are Klebsiella with ESBL expression. And our conclusion from this data here is actually there's no difference between wild type and non-wild type strains, and actually E. coli and Klebsiella could be described by the same target. The data sits quite on top of each other. So another thing that we really need to consider is variability. So often we explore PK/PD in more than one preclinical model. So we use the neutropenic mouse thigh model. We also use the lung infection model, particularly when we're developing for a pneumonia indication. But on top of that, we also use in vitro models, such as the in vitro hollow fiber infection model and the one compartment model. So this can result in huge variability in the PK/PD magnitude that we get from this data. And actually, in reality, it's not just using different models that gives you that variability. If you're just developing data from a single mouse model across multiple strains, you will get a large amount of variability that's associated with stasis and bacterial killing. And there are various methods of interpreting that PK/PD target. So when you have all of this data, you have to consider what you're going to use. Do you use the mean or the median? Do you use the most conservative value? Do you model all of the data together? Or do you consider all of the targets and use those in your PTA? I'm going to come back to these points later when talking about PTA to put a bit more information around this. So then we have to think about our site of infection. So generally we measure PK in the plasma. It's easier to get samples and we can get serial samples, particularly in human. However, we do need to consider whether plasma PK is always relevant. So particularly if you think about pneumonia indication, it's important for us to demonstrate that we get sufficient lung penetration. So one of the ways we do this is we measure PK in epithelial lining fluid as surrogate for lung tissue exposure. But if we're going to do this, it's important for us to understand that PK in the ELF, both in the preclinical model, in your mouse model, and in human. And what we're trying to understand here is that relationship between plasma and ELF similar across both models? Because we want to understand if it's relevant to use plasma PK. So if, for example, you demonstrate in your mouse model, you get 35% penetration into ELF, and actually you get the same penetration in human, and the shape of the profiles are similar, you could put forward an argument that the plasma concentrations are quite relevant. If, however, you're seeing a disparity in those relationships between the mouse and the human, you might need to consider the ELF exposure in more detail, because really it's only the exposure there at that site of inspection that you understand that will drive efficacy and could be translated. So the other thing we have to think of is emergence of resistance. So we know that we will have mutants likely to exist in the initial bacterial burden. And as we increase drug, that drug pressure will amplify the less susceptible population more. So actually you can get an increase in resistant mutants, and you need a higher dose to actually suppress those resistant mutants. So it's important for us when developing a drug to consider the target not only for bacterial killing, but also for suppression of resistance. And this plays in more why we have to use more than one model. It's very difficult for us in an in vivo model to really understand emergence of resistance. We need to run these studies out for longer, and we need to start off with a higher inoculum at the beginning. So a higher inoculum is not sustainable in an animal model, it will kill the animals. And running experiments generally longer than 24 hours, again, is not sustainable. Certainly you would lose your controls. So for this reason, we rely more heavily on some of our in vitro techniques, such as the hollow fiber or the one compartment model. And because it's so important to still keep in mind in your target that emergence of resistance, it demonstrates that you need to consider more than one model when developing your data. So now moving on to MIC distribution. Okay, here we go. Sorry about that. So we always relate our PTA to MIC distribution. So for our dose selection, it's important for us to understand what MIC we're wanting to target. So we generate data of MIC of the drug that we're trying to develop across many different isolates, panels of isolates. And ideally, what we're trying to do is develop a drug that's going to cover the majority of organisms that's expected in that indication. So ideally, you would like to have your dose cover the MIC 90 of that distribution. So in this example here, it's isolates of from pneumonia patients that have been treated, that have been tested in a susceptibility test with ceftazidime avibactam. And what the data is indicating here is the MIC 90 is around 8 mg per litre. And what you're, another thing to consider is when you're choosing your dose, you want to consider the least susceptible pathogen. So, you know, for a broader spectrum antibiotic, you'll have, so I think this might have been a Pseudomonas panel, but you'd have a Pseudomonas panel, an Enterobacteriaceae panel. You might look separately at E. coli and Klebsiella, for example, and it's quite likely that you'll get a different MIC 90 from the different pathogens. But in order to choose your dose, what you want to do is choose your dose to cover the least susceptible pathogen, i.e. the highest MIC 90. Other things to consider with this is you get more data on these susceptibility panels as you move through drug development. And for what you're using your PTA for, you want to consider the size of your surveillance. You know, there's about nearly under 1,500 in this pool here. But at the beginning of development, you will only have panels of maybe several hundred. So when you're considering your PTA and your target MIC 90, it's going to be more robust if you have a larger panel of surveillance that you're judging your MIC 90 from. You also need to think about whether you have sufficient representation of resistant isolates. I mean, it's very likely that your resistant isolates are going to have the higher MICs. However, if they only form a very small proportion of your total pool, if there's only 5–10% of resistant isolates in there, it's possible that your MIC 90 could actually not represent those resistant isolates properly, and you could get an under estimate of your MIC 90. As I was talking about before when talking about the PK/PD target, now when you understand your MIC distribution, you want to ensure that when you're evaluating your PK/PD target, you do include some of these isolates that are at the upper range of that target. Another thing to bear in mind is whether you have enough isolates from a relevant infection site. So the example here is isolates from patients with nosocomial pneumonia are considered to represent a generally less susceptible group of isolates than those from other infections. So if you were, for example, developing a drug for nosocomial pneumonia and you had most of your surveillance on isolates from UTI patients, for example, it's possible that you could underestimate your MIC 90. So moving on now to the population PK model. So firstly, what do we mean by a population PK model? So I'll go back to how we're measuring pharmacokinetics. In phase 1, we tend to take intensive PK samples. So we can take about 8 to 10 samples per subject, and we take them at very specified time points. So we can ask for samples at 30 minutes, 1 hour, 1 hour, 30 minutes, and potentially with a 5-minute window amongst it. And from this intense PK data, you can actually derive quite a nice concentration time profile, which allows you to calculate PK parameters such as Cmax, AUC, half-life, clearance, or volume of distribution. And it allows you to calculate that for each subject. However, when you get beyond phase 1, you start to get more limitations on how many PK samples you can take per subject. So generally you can take only 2 or 3 per patient. And also that sample timing becomes a lot more variable. So it's not possible to stipulate quite as specifically what time you want your samples taken. More we give a time window, samples need to be taken between 2 to 4 hours, because these are patients, often they can be patients in an ICU, and it's difficult to get those samples. So we tend to ask for something within a sample window, but that the time that is taken is recorded. So you can see here, if you just have these samples in isolation, it'd be very difficult to interpret the data because actually you could put any line through it. However, you have multiple of these samples from different patients, and also if you then pull in data that you might have taken from phase 1 and analyze the data together, you get much richer data for pool, and you can fill in some of those gaps and really start to understand the pharmacokinetics. And in doing that, we use that data, we pool all of this data together, and we build a population pharmacokinetic model. And what this model does is it describes not only the average patient, but it can describe the variability. And with this model, you can start to study and understand the influences of variability. So why some of these patients will have higher exposure or lower exposure. So there are many influences of pharmacokinetic variability. There's weight or body surface area, renal clearance, specific for especially for drugs which are predominantly renally cleared. The severity of illness can cause quite a lot of variability, and that can impact what on average pharmacokinetics can look like across different patient populations. So there can be differences between the PK in patients with hepatic hospital-acquired pneumonia or ventilator-acquired pneumonia compared to patients with complicated UTI infection. Age can have an impact, concomitant medications, and also ethnicity. So you can imagine that many of these variables are not present or restricted in standard healthy volunteer studies. Age, for example, unless you specifically study a geriatric population, you'll find that most healthy volunteer studies might be limited to subjects between maybe 18 or 40 years of age. And whilst you can study these in the impact of age and the impact of renal clearance, for example, in specific clinical pharmacology studies, they don't give as full a picture when assessed separately than when you pull all the data together. And particularly in those healthy volunteer studies, it's not going to be as heterogeneous a population as your patient studies. So you might not be able to understand the influence of someone with renal clearance and a high body weight together, for example, or the influence of age and renal clearance together. So because of that, it remains really important for us to continue to study PK in patients, even when we have healthy volunteer data. So the way we develop our population model is iterative. So we first tend to develop the population PK model based on phase 1 data in the healthy volunteers. And we use this data to model PTA analysis to select our doses for phase 2 and 3. But then once we get more emerging data from phase 2 or 3, we'll update our population PK model further with that data, rerun our PTA analysis, and potentially even modify the dose between these phases. So once we've developed that population PK model, we use it to simulate more patients. So we develop the model from hundreds of patients potentially, and then simulate what that data will look like, taking into account that variability we're seeing into thousands of patients. And we do this using Monte Carlo simulation. Also, now you've got a population PK model, you can use it to simulate different doses. And then the question we're asking from these simulations is, now we've got a full pharmacokinetic model from thousands of patients. And what we're asking is, what proportion of these simulated patients would have exposure above your PK/PD target at each MIC? And this is what we mean by probability of target attainment. So from this data, we generate our PTA graphs. So the graph that I'm showing here has two y-axes. So the PTA is shown on the x-axis here, and I'm showing that as a percentage here, or you can sometimes see it as a ratio of 1. And the x-axis is showing increasing MICs in doubling increases. So what we're looking at here is how the PTA changes with increasing MIC. So as you can see in this example, the PTA is close to or at 100 at very low MICs, and very slowly starts to come down with increasing MIC to a point where suddenly that PTA really drops quite sharply. And this is happening in this example at an MIC of 8. Now, to really put into context that MIC at which the PTA drops, we tend to overlay our PTA over that MIC distribution. So here we're reading the MIC distribution from the right-hand axis, so the percentage of isolates from our panel at each MIC. So you can see in this example here, your MIC 90 is probably around 8, and what you can see is the PTA is still high at that MIC of 8. So you could conclude from this that the dose that has been simulated here provides reasonable coverage for MICs up to and including 8. But again, I can't reiterate this enough. This conclusion is dependent solely on the robustness of that underlying data. You really have to have a good description of that PK/PD model that generates the PK/PD target, and you really have to have a robust population pharmacokinetic model and a good understanding of variability. So how do we use our PTA analysis? Well, we can use it in initial dose and regimen selection, and then we use it to continue to optimize that dose during our clinical development program. And particularly, we can use it to select doses or optimize doses for special populations. So for example, dose adjustments for patients with renal impairment, where the drug is predominantly renally cleared, or dose adjustments for patients with hepatic impairment. We can use it to select doses for pediatrics, and then other groups such as geriatric, obese, or different ethnic populations. The thing to bear in mind here is you're not only using PTA to suggest a different dose, but you're also using PTA to justify whether a dose is important or appropriate in these groups. So, for example, you might see in some of these categories, geriatric population, for example, you might see a slight decrease in exposure, maybe 10% decrease in AUC on average. But what you can do is use your PTA and look specifically at that group, at that age group, and assess whether actually you're getting sufficient target attainment. And you can use that potentially to justify that maybe you don't need a dose adjustment for that group. We can also use PTA analysis to support our breakpoint justification. So taking this together, we develop our PK model from all our clinical data. We conduct our Monte Carlo simulation, and now we're taking into account the demographic distribution of the target population. And then we put that together with the PK/PD target we've generated and the MIC target from the surveillance program. And we'll start to produce our PTA graphs. And generally what we're doing is we're looking at different things. So we're looking at our different doses and overlaying them on the same plot. Actually, sometimes we might want to consider different targets, you know, a target for stasis or a target for 1 log kill, and again, overlay the MIC, the PTA on the same plot. And what we're going to do is want to select a dose that is predicted to achieve high PTA at your target MIC. So when we're considering these simulations, we need to think of a few things. So in early development, as I've mentioned, generally our PK is coming from a healthy population, and we're using that healthy population to select a dose for patients. But as described, PK can alter quite drastically between patients and healthy volunteers. And certainly the severity of illness in the patient can have an impact. So what we can do when we're conducting our PTA analysis is inflate our variability so that we're not taking too conservative an understanding of dose through into phase 2 clinical trials. So an example of this is quite often we increase the variability on a parameter such as clearance, because we often see that clearance tends to vary a lot. We can also increase the variability on volume of distribution or other parameters depending on what you're understanding about the PK. So we identify covariates during the development of the population PK model, and we've talked about a few of them, such as creatinine clearance can be a covariate for drugs which are renally cleared. Age can be a covariate, and age definitely will be covariate when you're considering developing a drug for pediatric populations. So when you're conducting your simulations, you can specifically simulate in those groups alone. So you can simulate patients with renal impairment within a certain category of renal impairment. So you can separate out what the PK will look like for subjects who have a creatinine clearance which is more normal compared to subjects with moderate renal impairment, for example. We can do different simulations for different patient populations if that has come out as a significant covariate. So you could do a simulation that you believe will be more representative of your CUTA population versus your HAP population, for example. When we're doing drug development for pediatrics, we can simulate different age groups when we're choosing a dose. So here's an example of what this looks like. So this example is an assessment of dose adjustment for patients with renal impairment, and it's for a renally cleared drug. So here you've got your different categories of renal function. So normal renal function, mild impairment, moderate impairment. In this example here, I've not shown the doses, but there are different doses actually being simulated here. And this PTA is actually an assessment of if those doses are appropriate for each of these categories. But here, when that PTA is being looked at, it's being looked at separately for the complicated abdominal tract infection patients, CUTI patients, nosocomial pneumonia patients, and as a subgroup of that, VAT patients, because it's a subgroup that can have a bigger impact of especially creatinine clearance. But you can only do this if you've had sufficient data in your population PK model building, and these have come up as significant covariates to allow you to simulate patients with those different indications. So then we have to think about the threshold of the PTA that we use. Quite often we see talks about 90% PTA, and I think it might even be there in one of the EMA guidances. But is this 90% a magical number? Is 90% PTA high enough? It obviously means that it's possible that 10% of your subjects isn't going to get high enough exposure. Does it automatically mean that 85% PTA is unacceptable? Can you not develop a drug if the PTA is only 85%? And I don't have an answer to any of those questions because it's not clear cut, and I don't think you can have very definitive rules about it. It actually will only depend on the situation and the risk benefit. And also it'll depend on the amount of evidence. So for example, if you're using your PTA to justify a breakpoint, for example, you might need to set a high threshold for you accepting that PTA when you have less clinical data that is supporting that breakpoint decision. However, then if you have an unmet need and there's no other agents, or all the other agents have significant toxicity issues associated with it, you might accept a lower threshold. It might be more acceptable rather than not having that drug available. To put this into context, it's quite useful sometimes to benchmark to other drugs, such as the drugs that you're considering for use in standard of care or as your comparator in phase 3 studies. So I'm going to come back now to something I mentioned earlier in the presentation, which is the question of what PK/PD target we want to use for PTA analysis. So this is an entirely made-up example. But in this example, studies have been conducted in the mouse neutropenic thigh model, and 5 strains have been used, 5 E. coli strains have been used, and 5 Klebsiella pneumoniae strains have been used. There's varying MIC and varying, the strains are mixed up with in terms of wild type and those exhibiting resistant mechanisms. Bacterial burden has been calculated as stasis or 1 log kill. Generally, we'd look at 2 log kill as well, but I was just trying to make this easier to look at. And it's been assessed in both plasma and in ELF. And then the mean and the median and the min-max has been summarized here at the bottom. So here we have this problem of variability. So you can see from this data, with some of the groups here, you've got quite a large discrepancy between your median and your mean of the data. And you can see that there's a huge range in the data. So you have that question of what do you want to take as the most important target here? Is it the mean of the data? Is it the median? You'd probably consider it too conservative just to take the highest value. You've got to consider here that you've only tested this experiment against 10 strains, and you're comparing this to the number and the variability of your strains out there. You've had a bit more of an outlier in your strain. You can't ignore it totally, but you don't want to base everything on it, because again, you could throw out a perfectly good drug. What's happened in this experiment is that for each strain, the stasis and 1 log target has been calculated separately. So these are the values for each strain. So we could do it that way, or instead we could take all of this data and co-model it together. So rather than summarize a target per strain and get into this problem of which parameter to use, mean or median, you could co-model the data all together and say from all 10 of those all 10 of those strains, the stasis value is X or Y. Then you've got to consider whether you're going to use the plasma target or the ELF target. And I talked about some of the rationales for why you might use one or the other before. I mean, certainly if you can rely more on plasma than on ELF, you tend to have richer data from plasma simply because of the fact that you will tend to have quite rich data from your healthy volunteer studies and potentially quite a lot of data from your phase 2-3 studies in plasma. In ELF, even in healthy volunteer studies, for the majority of the time, you can only take one sample per patient or per healthy volunteer. And whilst you can do studies in patients, again, it's quite difficult to get those samples, and it's unlikely you're going to get it from your full patient population. So remembering that importance of your population PK model and understanding variability, if you have to rely entirely on ELF PK, you're not going to get as rich data. So it's good to understand both of these targets. Then there's the question of stasis or log kill and which is important. And again, I don't have a clear-cut answer to this. I think there's a general rule of thumb that stasis is possibly more acceptable for less complicated infections, and log kill is needed for more complicated infections such as nosocomial pneumonia, for example. But again, I don't think there are hard and fast rules around this. And this is another place where benchmarking can come in very useful if you're assessing your drug with the same criteria that you would assess what is currently standard of care. So everything I've talked about so far is considering the PK/PD target of a single drug, but what about combinations? And we're developing quite a lot of combinations at the moment, such as the beta-lactam, beta-lactamase inhibitors. And for these, we generate 2 PK/PD targets. So the target for the beta-lactam part of this is usually, for most beta-lactams, free time above MIC. So that's the PK/PD index. But the target for the BLI can be different. So it could be just an exposure threshold, such as free time above a concentration threshold or a threshold AUC, and not linked to MIC. But also there are some developments where that target for your BLI can be linked to MIC. So for example, it could be MIC over a multiplication of the MIC, a proportion of that MIC. However you calculate it, we need to consider the target attainment for both the beta-lactam and the beta-lactamase inhibitor. And when we present the data, we present that probability of target attainment against the potentiated MIC of the beta-lactam. So that's the MIC that is most important. And by that potentiated MIC of the beta-lactam, what I mean is the MIC of the beta-lactam when the susceptibility test is conducted with both the beta-lactam and the beta-lactamase inhibitor. So i.e. the lower MIC you get once the beta-lactamase inhibitor has been added. So you've got to consider both those targets, but there's also different approaches to how you consider that joint target. So one of these approaches is that you consider that target attainment is achieved if you can demonstrate that both the beta-lactam and the beta-lactamase inhibitor achieve their respective PK/PD targets simultaneously in each simulated patient. Another way that is being done is a stepwise process. And in this case, the PTA of the beta-lactamase is assessed first. And it's assessed against the MIC that's non-potentiated, so the MIC of the beta-lactam alone. And if the target attainment is achieved there, then you know that you don't need the BLI in that isolate, so you take as true the PTA of the beta-lactam alone. But if the beta-lactam target is not met, then joint target attainment needs to come into play, and that joint target attainment will be against the potentiated MIC. So your overall probability of target attainment is a combination of where the beta-lactamase inhibitor isn't needed and a combination of the MICs where it's absolutely critical. And both are valid and important approaches. So to try and put some of this into context, I'm just going to go through a case study. So I'm saying nothing new here, and I'm not even showing this as a particularly great and fantastic example of some of the concepts. It's just a concept to try and visualize some of the things that I've talked about. So the example is Zinforo, which is ceftaroline fossamil, a prodrug of an active component, ceftaroline. And this is a cephalosporin antibiotic with good activity against MRSA. It has been approved for the treatment of community-acquired pneumonia and complicated skin infections. And the efficacy was demonstrated by phase 3 non-inferiority studies in each indication. And it was excreted predominantly by renal elimination. So how PTA was used in ceftaroline development was that it was used for the initial dose selection for clinical trials based on that healthy volunteer data. And it was used to justify the doses for subjects with normal renal function and also the dose adjustments for patients with renal impairment. Data from PTA was used to support the breakpoint determination and used to predict doses for study in pediatric population. So it predicted, PK data was collected in pediatric population. Population PK model was built from this and used to simulate doses for efficacy studies. And then that was modeled again once those studies are being completed, and the PTA was used to justify the final label doses for pediatric population as well, which differed slightly from those studied in the clinical trials. Now, in this example, I'm just going to focus on showing you some examples from the Staphylococcus aureus data within the skin indication. So, to understand the PK/PD against staph, it was studied across a few different models. So there was the neutropenic murine thigh infection model. It was also studied in an in vitro single compartment model and the in vitro hollow fiber model. So there was a mix of models here. There was a number of isolates tested in each of these models. And from each of these isolates, a target for stasis, 1 log kill and 2 log kill was derived. And actually, there was quite good correlation across the different preclinical models. So in total, there were 24 isolates, and this included a range of MICs. You can see the MIC ranges from 0.12 to 4. And there was actually also a range of genotypes included within this. So there were both MSSA strains and also MRSA strains included. And from molecular characterization, there were slightly different types of MRSA isolates as well. The MIC distribution was assessed throughout the development program. One of the largest pools of data was from the AWARE 2013 surveillance study at the time that this analysis was done. And this included nearly 8,000 Staph aureus isolates, and nearly 4,500 of them were MRSA across different geographical regions. So really, just my take-home message here is you've got a really large proportion of MRSA isolates within this to really understand your MIC90 that you're trying to target for this drug that's targeting MRSA. The MIC90 was found to be 2 mg per litre in most regions, but there were a lot of geographical differences, and so it's another thing to consider and to take into account that you might need to look at this against different geographies of where you expect the drug to be used. So a population PK model of ceftaroline was developed. It was initially built with data from healthy volunteers, and then it was updated with data from patients with both CAP and skin indications. And the main significant influence of variability was creatinine clearance. So we developed that population PK model and then conducted the Monte Carlo simulations, and that took into account that demographic distribution of the target population. We took the PK/PD target that had been summarized across the preclinical models and took into account that MIC distribution from the AWARE surveillance program. And then PTA analysis was conducted for each dose for renal impairment, and it was assessed against MIC distributions across the different regions. So what this graph here is showing is the PTA analysis, and it shows the PTA analysis for both the stasis target in the round black star circles, 1 log kill by triangles, and 2 log kills, which is the I think the cross signs. And the proportion of the MIC distribution used, this is showing the European geographical location. So in this, out of that larger AWARE program, this is 5,380 isolates. The thing I want to just draw your attention to here is the impact of looking separately at the full distribution of isolates, but also it's important to look at those resistant isolates as well, because you might get a different understanding of where your MIC 90 is. You can see everything shifted slightly to the right. So I hope that through this talk, I've managed to demonstrate how PTA calculations are and can be an important and useful tool for dose selection and optimization, and also can be used for breakpoint setting. However, just to reiterate one more time, this PTA is only as good as that underlying data used to conduct the simulations. So what we need is that robust characterization of our PK index and magnitude. We need a really extensive assessment of our MIC distribution and a robust population PK model that has a good understanding, so that we have a good understanding of variability. And there's no hard and fast rules on how we use PTA and what target we actually want to achieve. It's really dependent on the circumstance, whether it be dose selection, breakpoint justification, or dose modification. It also depends on what data we had available for the choice at the time, and also the risk-benefit profile. So thank you very much for listening, and I will pass you back over now to, I think, Vincent.
Great. Thank you very much for your presentation, Shampa. So now we will proceed to the Q&A session. As a quick reminder for the audience, you can submit your questions as shown on this slide. We will try our best to respond to as many questions as possible during the allocated time. All right, so we have a couple of questions for you, Shampa, and I am going to start off with the first question related to infection model. You spent some time talking about the pneumonia model and the relevance of using ELF as compared to serum concentration. So the first question is, what about if we move on to an indication like a UTI model? Do you believe that the urine concentration is more informative than plasma concentration? Or do you believe that the PK/PD data that we generate from the standard lung or thigh model is good enough to be applied to UTI, urinary tract infection?
Thank you, Vincent. So in general, I'll take this in parts. In general, the mouse thigh infection model is considered to predict very well for urinary tract infection. So you don't need to necessarily have a UTI preclinical model. And actually, we believe that in terms of running PK/PD analyses, the thigh infection model is a bit more robust for that particular question. Now, in terms of what's the most relevant parameter, is it plasma or is it urine? Within complicated urinary tract infection, you've got to think of things like pyelonephritis as well, where you need some kind of tissue exposure. So we don't really consider just having high urine concentrations to be really sufficient. I think it's good to understand that you've got high urinary exposure, but really we will take our PK/PD analysis based on the plasma concentrations, and that's routine. But you will see quite often in drug development, as we're going through our justification and our understanding, it's still relevant and important to understand your renal excretion in patients and understand if there's high renal excretion.
Okay, so the second question is related to the use of MIC. I think that is more like a clarification. That could you explain further why is MIC 90 that is more commonly used and not just a specific MIC?
So I'm not sure if I can move my slides. No, perhaps I can't move my slides back. So I was going to just show you with the distribution. I guess what you're, let me go back to here. So if you look here, this is a really large surveillance over a number of clinical isolates. And what you can see is you want to use this drug for the majority of the population where there's a problem. And what you can see from this distribution here is that if you target your MIC90, you're actually capturing the majority of isolates there. And that's what you're developing your drug for. And that's why we're trying to go for the MIC90 rather than something in the middle. Another thing to consider is when you're selecting dose and when we're considering clinical breakpoint, and quite a lot of the breakpoint committees worry about the wild type population and not having a dose or a breakpoint that sits within the wild type population, because that just reflects variability in MIC testing. And so ideally you want to be at this end of the population where you're starting to see a difference.
Okay, so the next question is related to the timing that we need to do population simulation. In your presentation, you seem to imply that we need about 100 patient PK data in order to be able to model what is going on, what is likely to happen in a larger population. And that seems to be a lot of data. Could you comment on what is the optimal time that we should be using, we should be engaging this population simulation?
Oh yeah, and sorry, that really was just an example of where you might get to at the end of phase 1 if you consider all of the clin pharm studies. But no, you can start to build your population model from very early data sets. So sometimes we even start to build it during our SAD/MAD studies. So by the end of your MAD study, you can have a very early basic population PK model, and that's going to start to inform how what kind of doses you might want to take into phase 2. You might even want to model your single ascending dose study to actually model what kind of multiple dosing you want to have a look at if you've got time to do it. So you can still build data from smaller data sets. You just need to consider how robust that model is at that time. I mean, certainly something built from a much larger data set is going to be more robust. But that's why I mean it's an iterative process. You continue to build a model and then update it with more data again.
Okay, so the next question is interested in your opinion of using targets that include parameters beyond the MIC. So an example was given using Newton selection concentration as a alternative way of targeting what is how much you need. Could you comment on that?
Yeah, so really so let me just go back to the slide where I'm showing this. So it is important for us to consider the longevity of the drug that we're trying to develop. We know that when we develop a drug, some of the newer drugs that have been recently developed and being used, that have been licensed within the last few years, you're already starting to see emergence of resistance to them. So all I'm showing here is, we can run experiments where we're looking at that bacterial killing, but we're also measuring in these experiments the population of resistance mutants. And what we want to do here then is actually dose to a point where we can suppress that population of resistant mutants and base our target there. But we have to balance that target with what we understand also with bacterial killing and from the in vivo models. If we're too dogmatic about this, you can start to, say your target needs to be so big, you're never going to be able to develop that from a toxicity point of view. So that's why you have to balance these different data together.
So the next question is really on the model selection, the animal model selection. So in your presentation, you really mentioned a lot about the neutropenic mouse type model and also the pneumonia model. This is pretty much the standard models that we use. So the question is related to what about an immunocompetent model, okay, that we use, and also studying the drug concentration at the effect site rather than just the systemic exposure. And the specific indication that the question is interested in asking is about bone and joint infection. Are these useful to be part of the package for the regulatory agency?
Yeah, I think we tend to use different models for different reasons. There's been a lot of work done on the thigh and the lung infection model, and one of the reasons we use it in neutropenic models is because actually you can start to clear your bacterial burden by the immune system alone, and you get a lot of variability with it. So actually by using a neutropenic model, it's a bit of a cleaner system, and you're only looking at the impact of the drug. And really, we're just looking at the PK/PD. We're just wanting to have a clean system, and we're looking at how the exposure of the drug impacts the response. So when you use a non-neutropenic model, for example, immunocompetent model, for example, you'll get a lot more variability, and you'll be also looking at partly the impact of the immune system on clearing of the bacteria that might or might not be related to the concentration of the drug, so you might not get quite as clear a picture. I'm not as familiar personally with some of the models used for joint infection, but again, the mouse thigh and lung infection models tend to be used because they're sometimes less complicated, less severe than some of the other models, like for sepsis, for example. So they're a better place to understand PK/PD, but they're useful to look at hand in hand with some efficacy data from some of the other models, like a sepsis model or the actual UTI model itself.
All right, so the next question, we are going back to the drug exposures in ELF. So is there any clinical evidence to support that ELF drug exposures are more important than plasma exposure for serious lung infection that may involve lung tissue and not just the airspace? That is, when penetration studies in mouse and humans show discordance, how confident are we that we can translate the information from the mouse ELF to humans?
So let me just check if I've understood this. So there is evidence that lung penetration is important. There have been clinical phase 3 failures in nosocomial pneumonia, and through investigation of this, there seemed to be a huge discordance between what was understood from the mouse and what was understood in humans. So certainly, it is considered to be important to understand penetration at site of action. If there is discordance between your mouse and your human, if you measure your mouse ELF concentration, if you think about what we're measuring there, we're measuring of that concentration of drug directly on the bacteria. So we're setting a target on specifically that site of action. Then yes, it is a bit of a leap of faith, but then you're considering that if you have human ELF concentrations that are the same, it should have the same effect. And certainly, there have been recent studies where ELF has been understood as well as plasma, and they've resulted for selection of dose, and it has resulted in successful phase 3 studies for nosocomial pneumonia.
So this is kind of a 1-2 punch question as a follow-up to the previous one. So I'm going to skip the next two and then go straight to this because this is more related to the one that you just answered. So what is the potential of using physiological-based model? So this is the PBPK model in order to simulate patient PK that would address site penetration in addition to patient variability. Is it a feasible way to estimate PTAs?
Yeah, I think it's feasible, and I think there's been a lot of development and work in this area. I think it's just, there's a lot that's not really fully understood yet with the physiological based modeling on how well and how accurately it predicts. You need a good set of data to confirm that the model is predicting accurately, and we don't always have those loops. So whilst I'm seeing it as a growing field, I've not seen it in those final regulations and dose decisions made as frequently yet. But I think this development's going to continue in that area.
Okay, so let's go back to the line of the question. So this one is, which one is better, one compartment model or the hollow fiber model? When would you use? When would you use which?
So You know, I think they're used for different reasons. You know, I see an advantage of the hollow fiber model. You know, I'm biased. I'm the wrong person to ask this question because we run the hollow fiber model within our lab. So I can't really give you an open answer. I can just say that it's a system where, you know, at least you've got your bacteria growing on your hollow fiber tubules, so you've got some separation. You can run this system out for several days. You can sample the system. You can look at emergence of resistance. You don't have the mixing problem. You're not diluting the amount of bacteria in the system. So I think there are certain advantages of it. I can speak less to the advantages of the one compartment model simply because I don't personally use it and don't have as much information on it.
Okay, so the next one is about how do you decide on optimal PK sampling time? So this is optimal sampling for rich and sparse sampling studies.
We often do that by simulation. So we can use simulation. So we have our population model that's been developed with our rich PK data. And then you can simulate what it would look like if you use different sampling schemes. And then you can choose an optimal sampling model from that. And that's used quite frequently. I think there's particular programs that you can use to do that.
Okay, so this one is, the next one is kind of by intention. So I'm just going to read out the question first. You didn't really go too deep into the beta-lactamase inhibitor combination example. Would you comment on how would you progress on these combination starting from the PK/PD studies to PTA?
So I'm not sure I understand the question. How would I progress from? So tell me the question again. I'm not sure I'm getting the gist of it.
All right. So you didn't spend too much time detailing the details on the BL-BLI combination. Would you comment on how would you use this combination starting from PK/PD studies all the way to the PTA estimation?
Sure. I mean, that's a long and complicated story in itself. So when you're looking at the original, you know, PK/PD target and driver, it's much more difficult than looking at a single agent on its own. So what you're trying to understand is the target and driver for your index and magnitude of your BLI. Generally, the beta-lactam is understood already. And at the moment, I'm going to give you an example where we're assuming these two things work in isolation, that the beta-lactamase inhibitor doesn't have antibacterial properties itself. So when you're running your dose fractionation and when you're running your magnitude studies, you've got to be able to select a dose of your you can only do this in the presence of your beta-lactam, because remember, your inhibitor doesn't have activity itself. But you've got to then select a dose of your beta-lactam to test in combination that's not going to be doing the work itself. So it's got to be at a level where there's sufficient beta-lactam there that in the inhibitor's doing its job, you're going to see an impact of the inhibitor, but not too much beta-lactam so that you're just seeing the impact of the beta-lactam alone. So you need to find this precious space where you're really investigating the beta-lactamase inhibitor alone. So I can tell you that takes a lot of experiments, a lot of hard work to actually select those. And you have to be very selective sometimes with the strains that you're using to do it. And in these experiments, now we're focusing only on resistant strains because obviously you're looking at the impact of the beta-lactamase inhibitor. However, once you've done that, it's quite similar to how you're studying a compound on its own. You might want to do some additional work on understanding the index and magnitude of your beta-lactam, but quite often, you know, with the recent drugs that have been developed, that is already very well established. So, for example, we've seen combinations with meropenem or ceftazidime or imipenem, and those are very well characterized drugs, and we already understood the index and magnitude of those drugs. So now we know the index and magnitude of our beta-lactam. We've generated an index and magnitude of our beta-lactamase inhibitor. We then go, so that's the PK/PD target. When I was talking about the MIC distribution, some of the MIC distributions I actually showed in my examples. I don't think I can get back to it. were from ceftazidime avibactam. So the MIC distribution that I was showing was from a panel where ceftazidime and avibactam being tested in the susceptibility testing. So here you've got ceftazidime with a fixed concentration of your beta-lactamase inhibitor. Now, I'm struggling with the opinion that the concentration of the beta-lactamase inhibitor is not related to the concentration, that PK/PD target that you've generated in a fully dynamic animal and in vitro based model. So you've got then your PK/PD target of both components. You've got your MIC distribution, which is one MIC distribution based on the potentiated MIC. And then you build your population model. So what I've been involved with recently was ceftazidime avibactam, and what I've seen published for some of the other combinations is generally two population models which are describing, the beta-lactam and the beta-lactamase inhibitor. But then when we're doing that PTA analysis, what we're wanting to do is simulate that for each patient that you're simulating, that patient is achieving both your beta-lactam target and your beta-lactamase inhibitor target simultaneously, and that's what's going to form your PTA. And then I talked a bit through an example of how that's considered sometimes slightly differently in a more stepwise approach. Does that start to answer your question? Did that go through the process?
So there is also a follow-up from the same person that with this kind of analysis, would you be able to distinguish whether the BLI is microbiologically active or not? How would you distinguish from the PTA analysis?
So I don't think you can distinguish that from the PTA analysis. So in the way I've just described it, you're assuming there's no biological activity of your BLI, so no microbiological activity of your BLI. I think you might have to assess it very differently if you're looking at, so there are some BLIs being developed at the moment which have their own intrinsic activity. And so you'd have to go back and consider that within your target. In how you're thinking about your target. I personally haven't worked on yet a beta-lactamase inhibitor with intrinsic activity, but I think you will have to consider your target slightly differently.
Okay, so the next question, let me go back to the line, the sequence that I was looking at. All right, so what is the difference between an isolate and a strain? This is the first part. And what is the minimum number of the isolates that you need for a robust PK/PD PTA analysis? I get this all the time.
Yeah, I don't. So first question, I'm using these words all interchangeably, and I mean each strain that you're using in your experiment. So I'm calling it isolates. Sometimes I'm calling it strain. Minimum number, nobody knows the golden answer to this question. I would say that when people come to speak to us about, supporting them with a pre-KPD package, we say that really to understand it, you want to understand it across at least 10 to 12 isolates. If you're talking about something that's quite broad spectrum, that's going, Enterobacteriaceae, quite often we see E. coli and Klebsiella data sitting more on top of each other. But if you're then looking at Enterobacteriaceae and Acinetobacter and Pseudomonas, you want to make sure that maybe that full number goes up a bit, because you want to ensure that you understand each of those pathogens quite well. But there isn't a golden rule. There isn't a good answer as to what that particular number is, to be honest.
Okay, so the next two questions are from two different attendees, but practically they're asking the same question. So I'm trying to paraphrase the question so that I can get the idea across. So should PTA analysis consider variability in MIC? Meaning that MIC is a static parameter that is set up with twofold dilution, and there could be variability depending on the day, depending on the condition, that you could have variability on the estimate of the MIC. How can we incorporate this inherent variability in MIC determination into the PTA analysis?
I think I think that's a difficult question. I mean, I think there's probably methods to do that. So let me answer a very slightly different question, and I wonder if it will answer the same question. One of the things that I showed here was how PTA, I presented PTA per MIC level. The other way you can present and think about your data is PTA over the whole of the distribution. So your overall PTA is not just your PCA at your MIC of 2, but it's your PTA taken over the distribution that you're expecting to see. I think if you start considering your analysis that way, you could possibly also include variability on MIC technique in that. But personally, I think that's quite a muddy field. We know MIC analysis is variable, but I think trying to understand PTA and include MIC analysis variability is going to get very complicated.
All right, so I think let me just see if there are any more questions coming through. I think these are the key questions that we have gone through. Okay, so great. So now, thank you for staying. We now have come to the end of our Q&A session. I'd like to thank everybody for the questions and comments. And once more, thank you to you, Shampa, for the excellent presentation.
Thank you.
Thank you very much, Vincent, for moderating today's webinar. And of course, thank you very much, Shampa, for your presentation and for sharing your expertise with our audience. I'd like to take this opportunity to announce our next webinars in May, June, and May. We will hold our webinar in collaboration with the Pew Charitable Trusts on assay development for measuring antibiotic accumulation in gram-negative bacteria. On 9th June, Vincent Tam will also present his own webinar on the topic of PKPD or fixed dose BLBI combinations. Marcus Zeitlinger will present on phase 1 development challenges in the clinical development of antimicrobials on 30th June. And on 9th July, Anand Anandkumar and Kamini Walia will present a webinar on the challenges and opportunities for antimicrobial R&D in low and middle income countries. You can already register for these webinars on the Revive website via the link shown below. So that will be all from my side. I'd like to thank everybody for joining today and for contributing to this discussion. I really hope you found this webinar interesting and useful and that you'll join again in future. Please do make sure to spread the word in your networks and to encourage your colleagues to join as well. Thank you and goodbye.